In today's video, we're going to be taking a look at the structure of roots, stems, and leaves in dicotyledonous plants that we need to know for AS biology. If you want to skip to a certain organ, take a look in the video description below as I've listed the respective timestamps. Otherwise, let's go ahead and get started with the roots. So, roots have their vascular tissue arranged centrally, right? It's located centrally in the organ, with the xylem being star-shaped and the phloem tissue being separated by the tips of that star-shaped xylem. We then have a pericycle, which is made of parenchyma cells, and we then have an endodermis. Now these here are a few endodermal cells, and we see that they have these regions shaded in, and those regions that are shaded are called Casparian strips, which are made of suberin and are waterproof. We'll get back to that a little bit later on. We then have a cortex, which is made of parenchyma cells, so packing tissue, and then we have an epidermis, which has these protrusions called root hairs. Now, root hairs are responsible for the uptake of water and mineral ions. So when the water and mineral ions enter via the root hair, they pass through the cortex, and the mineral ions are dissolved in the water. And when they reach these endodermal cells, the water and mineral ions dissolved in the water reach the Casparian strips, and because the Casparian strips are waterproof, water traveling via the apoplast pathway, so in between the cell walls, has to go into the cytoplasm, right? It diffuses into the cytoplasm via osmosis, then it passes through the pericycle, and then enters the xylem via the apoplast pathway. Stems have vascular bundles arranged in a non-continuous ring shape. Now the structure of a vascular bundle from a stem is as follows. So they have sclerenchyma towards the epidermis. Now sclerenchyma is composed of cells which have thick cell walls. Normally they're lignified, so they're very strong. We then have phloem, we then have cambium tissue, and then we have xylem. Now cambium is a meristematic tissue, so it can differentiate into either phloem tissue or xylem tissue. Now, the reason why the vascular bundles are arranged in this non-continuous ring shape with sclerenchyma towards the epidermis is because stems are subject to a lot of outward pressures like strong rain maybe or wind, and in order to be able to withstand those forces they need to be strong, and this arrangement with the non-continuous ring shape and the sclerenchyma provides the strength required. We then have a pit, which is made of parenchyma cells, so packing tissue again. And then we have a cortex, okay? Now the cortex is composed of parenchyma cells, so packing tissue, and collenchyma. We can see it here. Now collenchyma is a strengthening tissue like sclerenchyma, but collenchyma is found in areas of growth within a plant. And then the outermost layer of cells, the outermost layer of tissue is called the epidermis. Now in leaves, the vascular bundles have the xylem on the top and the phloem on the bottom, which is the exact opposite to how it is in stems. You have to remember that in leaves it's the xylem on the top and the phloem on the bottom, and then in stems it's phloem on the top and xylem on the bottom, and then we often have this, we have this cambium tissue. This here is a large plan diagram of a leaf. So we can see here we have the upper epidermis, we see here that we have the lower epidermis, we have our vascular bundle here, and we have this line, which I'll come back to a little bit later on. So firstly, let's take a look at the vascular bundle. So we have collenchyma tissue, we then have xylem above the phloem, right, xylem above the phloem, and then we have some parenchyma tissue. Now in this space beneath the vascular bundle, we have collenchyma. I've drawn the layers of cells within a leaf. The topmost layer is the upper epidermis. The layer beneath that are the palisade mesophyll cells, which are these long elongated cells that are very tightly packed to maximize light absorption, to maximize photosynthesis. Beneath the palisade mesophyll cells, we have spongy mesophyll cells, which are very loosely packed, and that provides a lot of air spaces for where gases can diffuse into, like water vapor during transpiration. We have guard cells, which have pores called stoma. And through these pores, through the stoma, is where gas exchange occurs in leaves. Now coming back to this line, so I've drawn it to recognize that we have palisade mesophyll tissue above and spongy mesophyll tissue below. So that's it for this video. If you enjoyed, take a look at some of the other content on my channel that you might find interesting. Otherwise, thank you very much for watching, and I hope you have a great day.